I hope you can see my slide, right?
Yes, sir.
Yes.
Yes, sir.
Very good. So, as you can see in the first slide, even left side, you have some like beads like structure that are joined together. This is just amino acid sequence. This is called the primary structure of a protein. That means whenever the protein is just getting translated, after that it looks like this. But in order to function, it needs to fold. On the right hand side, you can see some kind of simulation based study where we are showing that how a protein can fold from its primary structure. Primary structure means just the order of amino acid sequence to some kind of three-dimensional structure so that it can function properly. So every protein, they have their own three-dimensional structure with specific properties so that they can carry out particular function. Here I would like to mention, like I told you before, that say for example DNA, whether it is bacterial DNA or human DNA or DNA of some human, some monkey or other organism, the basic architecture is same because the major function is also same. They are just storing information. And from DNA to RNA, RNA to protein, all those things are very I would say very conserved kind of thing. And DNA looks like same everywhere. But now the thing is, from that information, when you are getting proteins, say for example in human more than 1 lakhs of proteins, right? But those proteins are actually completely different to each other. Different means in terms of their overall organization and three-dimensional structure, so that they can perform different types of work. Like some of them are polymerase, some of them are digestive enzyme. So all of them have different types of catalytic activity if they are enzyme, and some other proteins are structural protein. They also help in different types of support in our body. So in order to perform specific function, they are different. That's the bottom line. Okay. So here, this is just refreshing the old thing. So I'm not going through it, but just I want to point out by this time, all of you know that what is codon in RNA. So codon means three-letter nucleotide present in mRNA. Uh, what
So
happened?
please start recording.
Huh?
No, sir. No, sir. Nothing. Nothing.
Okay, so please unmute here. Okay, mute. So here, so codon means that's three-letter code in mRNA. So here, from three-letter, from one codon equal to one, amino acid. So that way, finally, you will get your protein. Those are the molecular machine, and we call like polymer of amino acids. So this is very simple. So here, from here, like something new. So as I told at the beginning, that proteins come in various shapes and sizes, right? Here you can see this is hemoglobin. All of you know what is hemoglobin. Hemoglobin carry oxygen in our body. And all of you know where hemoglobin stay. So hemoglobin present in red blood cell. One thing you might not know is the number of hemoglobin per cell. In each of those RBC or red blood cell, approximately 270 million of hemoglobin is present. 270 million of hemoglobin present so that they can carry oxygen and support us. Okay, but now if you see like how hemoglobin looks like, they are kind of globular protein, and today by end of this class you will understand how from a primary structure, primary means just amino acid sequence, from there how you are getting this compact three-dimensional fold so that it can achieve its particular function. Okay. And another category of protein, we are just showing some of the examples. There are different types of proteins in our body. So here, this is some kind of cell membrane, and in the membrane itself, you will find different types of channel, like voltage gated channel, ligand gated channel. So all those like detailed informations at this point you don't need to require, like you don't need to remember. But here what I want to say that their function is completely different from hemoglobin, right? They are just channel and they transport ion in different direction, either from cell to outside or outside to inside, right? So the third one here you can see, this is called tubulin. So tubulin is very common, present in every cell in our body, right? So hemoglobin is present in red blood cell, but tubulin is present in every cell. So you can see here, it looks like some kind of hollow tube, right? Inside is hollow. Yes, that is true. And here, this hollow tube made up of two proteins, and they are present in repeating units. So here you can see that one yellow and one brown, yellow, brown, kind of repeating subunits, right? And they form this hollow tube. And this is absolutely required, like in our cell. So those are actually some kind of, they are providing like these proteins are called kind of cytoskeleton. That means they help to maintain proper shape and size of a cell, as well as tubulin, they help, or they are the intercellular tract inside the cell. Intercellular tract means, so for example, you are going from your hometown to some other place, right? You can take some kind of railway system, some train. So they have some track. So inside the cell, different things are moving from one direction to other direction. So all the time, those things are not happening randomly. Some track systems are also present in our cell, so that some particular molecule can go from this side to that side. So cells are very small, right? But inside the cell, those macromolecules, those molecules are even smaller. So they need to go in different direction at proper time, right? For that, there are a lot of mechanisms involved. Tubulin is one of them, and its major role is just like a track. So in the next diagram, you will see that this one. See, this is again some kind of computer simulated diagram, definitely video. So here you can see this portion, this tubulin, this is same here. You can see little bit of tubulin here, the same thing here. And now this thing is happening inside a cell. So that means we are just zooming we are showing in large scale, but that's all. So here you can see this is a vesicle. Vesicle means like inside the cell you have small vesicle. Those are some kind of cargo. So they carry, as I told, that they carry some cellular materials which should be transported from this side to that side, something like that. So those are the cellular vesicle. And you can see this person, right? This is nothing but a protein. This pink colored stuff, you can see it looks like it's walking through the track and holding this vesicle by its hand, right? Yes, this is true, but this is just a protein. Again, this is called motor protein. So it is functioning in that or in such a way that this vesicle can be transported to from one direction to another direction, and this is not in a random way. There are some logic and some mechanism like by which this direction is maintained inside our cell. Now, whatever I discussed in the previous slide, just to give you an impression that proteins are of different, level, they have different architecture and they have different function. But all of them, they should be started with the simple thing, nothing but polymer of amino acids. We have 20 different amino acids in natural condition, right? 20 amino acids in our body, and they are actually attached through peptide bond, I told you before, and that makes polypeptide, right? Polypeptide, and when it's folded into its three-dimensional structure and it has some specific function, that is protein, right? Now, why they are called amino acid? Most likely all of you know, because their like repeating subunit or their monomers are amino acid, right? So they have like one amine group and one acidic group, right? So one amine group and one acidic group. That's why they are called amino acid. And their basic, their overall structure is same. Like they have one central carbon, this one or C alpha atom, and they have one hydrogen atom attached to this central carbon and the R group or the side chain. So this R group is different in 20 amino acids. Why we have 20 amino acids? Because we have 20 different side chains. So R groups, normally I will refer it as side chain. Okay. So whatever I discuss, you will find everything here. So this is the central carbon, and this is the carboxyl group and amino group, and based on the pH, its ionization state will depend, right? So that you understand. So all of you studied chemistry before, and you have this knowledge. So initial part, I will go little quickly. These things I believe that you have proper idea, and when it will come to details of protein, there I will spend more time. Okay. And in the meantime, as all the time I say that if you have some question which are relevant, you can ask me. So here 20 amino acids that constitute a protein. So here you can see, again, I should mention that though these are the chemical structure of 20 amino acid, but you don't need to remember this structure. Rather, you should concentrate that we have different types of amino acid based on their side chain, because other things are fixed, right? This COH, NH2 group, H, those are the fixed thing in every amino acid. But this is the variable thing, side chain. So based on the side chain, we categorize this amino acid in different groups. Say for example, this group of amino acids here, you can see they have non-polar side chain, or you can say hydrophobic side chain, right? Non-polar side chain. So this group of amino acids, they have polar side chain. So you can see CH2OH, here also you have OH group, here you have SH group, all are polar group, right? So now this is positively charged R group. They are also you can say also polar, but they are positively charged. So positively charged means where in their side chain itself, right? We are not considering this amine or carboxyl group. Apart from that, their side chain is itself positively charged. Only 3 amino acids are there: histidine, arginine, and lysine. You don't need to remember their structure. And there are 2 amino acids which are negatively charged, right? This glutamic acid and aspartic acid, and some of them, they have aromatic side chain. So here the bottom line is, as I told you, nothing you need to remember here. The only thing you should remember is that glycine is the simplest amino acid, where the R group is nothing but one hydrogen. This is the R group in case of glycine, the simplest amino acid. And now if you can increase the complexity here in the side chain, then you are getting many things, right? Many amino acids. But we roughly categorize this side chain or R group into two broad categories. One is hydrophilic, polar, and the other one is hydrophobic or non-polar. Why I am telling this? Because this is very important to understand how protein folds. Because you can understand after synthesis of a protein, it's a linear chain, right? Now, if some of those amino acids are highly hydrophobic, their side chains are highly hydrophobic. So they will not be stable in solution. Solution means in our cell. So what is the solvent in our cell? It's aqueous, right? So as a result of that, in aqueous condition, in our like cell, inside our cell, those linear chain of amino acids, they will not be stable because they have multiple amino acids which contain hydrophobic side chain. Hydrophobic, from the name you understand, they are in fear of water. Hydrophobic, right? So as a result of that, protein needs to be folded. So it will form some kind of compact structure where hydrophilic side chain or hydrophilic residues will be on the surface and hydrophobic amino acid should be inside the core so that they can avoid water molecule. So as a result of that, it will give you a more stable structure. But not only the stability, in addition to that, it should fold in a proper way so that it can function, right? So all those things should be taken care of, and then your protein is functionally active, properly folded protein. Do you have any quick question? So it's clear, right? Whatever we are discussing.
Sir, can you please repeat the hydrophobic part?
Yeah, hydrophobic part means, if you see the structure in the side chain, you will see like they are not polar group or charged groups are there. So as a result of that, in water, they are not soluble or they are not liking water. So those are the hydrophobic side chain. So if they are also present, like since those amino acids are almost randomly distributed in a protein, right? If you just initially you just think in that way. So as a result of that, if the protein is linear chain, like then those hydrophobic side chain will create problem. So as a result of that, protein needs to be folded so that these hydrophobic side chains, say for example here, this is tyrosine, phenylalanine, tryptophan. So those side chain will be packed inside so that they will not face water. And as a result of that, the molecule will be stable. That's the bottom line. But I will discuss this thing again in the folding section when I will discuss more about this. So now, formation of peptide bond, I'm not discussing this. All of you are aware about this. It's okay. So, and now here.
Excuse me, sir.
Yeah.
Sir, the hydrophobic part is basically like non-polar. So why do we need to protect that part?
Because so what do you think that if hydrophobic, right? So that side chain, they will not actually, they will not be able in stable in solution. So for example, you have one linear chain and multiple hydrophobic amino acid. All of their side chains are hydrophobic, right? So if, say for example, if some polar group there, they will form some hydrogen bond with water molecule, right? So that way they will be water soluble. They will love to stay with water. But hydrophobic, they will form any hydrogen bond with water molecule, or they will be happy. So is it clear, Rajesh?
Yes, sir. They will not form bond, but why do we name it hydrophobic then?
Yeah, hydrophobic because as I told you that since they are actually, if you if you, I would say if you just synthesize this thing, not in terms of natural condition, if you synthesize that like some kind of linear thing, like because of this side chain, like whatever we are telling hydrophobic, like this linear chain, this hydrophobic side chain, they are not making any polar contact with the water molecules. So as a result of that, this protein naturally will tend to fold together. And when they will fold, this side chain will go inside so that they are not facing water anymore, right? So that's why they are hydrophobic. That phobia means in like fear, like phobia, and hydro means here water molecule, right? So if you see all of those side chain that contain either OH group or some charge group, those are the hydrophilic one, right? And most of those aromatic group or just methyl or CH2, CH3, CH3. I'm not telling exact amino acid name, that is not required. Those are like hydrophobic side chain. Okay.
So, like, is this due to the fact that water somehow neutralizes or maybe stabilizes the hydrophilic structures and not the
Yeah,
hydrophobic
definitely,
structures?
definitely. As I told that if you see like any structure, so at the end you will see, actually I will show you that like water means nothing, right? Water means they are constantly making hydrogen bonding, right? Even just water molecule if you see. So as a result of that, like any, like for example, lipid bilayer, I think in school days you also see lipid bilayer, how they form. They form like layer because they also have like hydrophobic side chain, like not side chain, that is kind of fatty acid chain. But again, they are staying away from the water, right? Because they cannot form like some kind of contact with the water molecule, and that's the problem. Because water is a polar solvent, right? So that's why that's the problem, I would say.
Okay.
Yeah. So now,
Okay, sir.
yeah. Now this two slide, I will go little fast, but if some of you are interested, I will suggest you to read little bit more. This is little like advanced like topic, I would say, in this level, but uh, this contribution actually has been made by one like Indian scientist who is like very famous in the field. I will introduce him also, right? But like very much of details you don't need to go right now. But some of the student like who like want to like pursue higher study. Say for example, particularly in physics, I would say if you are studying biophysics, this is like definitely you have to read at some point of your time, right? So actually, this has been discovered by Ramachandran, who was a physicist in IISc Bangalore. And I will tell more thing about this because this is actually important in terms of protein structure. Okay, so here, if you see, just very like it's almost like a storytelling way, I would like to introduce this. So if you see here, this is like primary structure of an amino acid, like of a polypeptide chain. So here you can see this is one amino acid, this is the central carbon, and here the R1, the first amino acid, side chain 1. So then you can see here, as I told you, that alpha carbon atom or central carbon atom is attached to one COH group. So here CO after the peptide bond formation, that OH actually is not there anymore. So you have the peptide bond, right? So before that, I believe all of you know that one water molecule will leave and you have this peptide bond here, right? CONH, finally you have this. So now this is the peptide bond, and then again you have another alpha carbon or central carbon. Then again you can see another peptide bond here. So it will be continued like this. Now what I want to say, because of this CO and NH group, this bond is it has some kind of partial double bonded nature, double bond character. And this bond is not that much flexible also in terms of angle, I'm saying dorsal angle. So this is also not flexible. But if you see the next two, like here, so from this figure you can come just in the bottom line here in the figure mentioned in the below. So this NC and CC, those are the torsion.
Excuse me, sir.
And,
Sir, can you zoom out the slide? We are not able to see completely.
No, in the bottom nothing there actually. Just whatever that N is bonded with something, it's not required. This is H here, so it's not required. C here you have O here, right? It's not required.
Okay,
Here
okay.
just it should be fine. So N
Okay.
C, this CN, this is the peptide bond. Like here, like in all protein, like we mentioned, it's omega. This is relatively like it has no, like I would say it's rigid, and then these two are flexible. like NC and this CC. This is called phi and psi, and then again omega, and again phi psi, again omega, again phi psi. It will be continued like this. So since I'm mostly I work in this field in protein structure, function, protein engineering, but I would not like to go in the details. Whatever it has been, it will be covered in all other section. I will just discuss at that level. The idea here is that, so if you have like one linear chain of polypeptide. So you have one amino acid here, another amino acid here, like it's going like this, and those are attached by peptide bond, right? So when the peptide bond is formed, if you see the previous slide, so as a result of that, at one terminus, like at the here, you have the H2N or NH3 plus, the amine group, and at the end you will have the COOH or COO minus. This is carboxy terminal at the end of the protein, right? So as a result of that, in DNA, RNA, you are talking about 5 prime, 3 prime, 3 prime, 5 prime, but here no 5 prime anymore. We say N terminus, that means amino terminus, and here C terminus or carboxy terminus. How it formed? Just see here. Here you have amino acid number 1 and amino acid number 2. When they make peptide bond, you have here NH3 group, here carboxyl group, right? So add another one, sorry. Add another one here, third amino acid. So it will be added, okay. And then again, you will have COO minus here. So again, you have to add another one, right? Where it is happening? During translation. As I discussed during translation class, like peptide bond formation will take place inside the ribosome. Ribosome will help that. And finally, you will get a linear polypeptide chain that contain at one end amine group. That's why this is called N terminus. And here C terminus. Some protein can be 100 amino acid long, some might be 500, 1000, 5000. There are many proteins of different or various length, okay? But they need to be folded correctly. Then how this folding will happen, that is big question. And many people across the world, including biologists, biochemists, physicists, they are working. Many things are known, but many things are unknown also, how protein folds. Why it is important? Because of some misfolding, there are multiple diseases actually happening. For example, many of you heard about like Parkinson disease, Huntington disease. Like there are a lot of diseases which are because of misfolding of protein. So as a result of that, understanding protein structure, how protein folds is very important. And people are working with this problem for a long time, and we have a lot of solution also nowadays. But still, let us discuss about the very basic thing about proteins. Now, what we started, that this phi and psi angle, these are like basically torsion angle, and now this phi, psi, those things are introduced by Ramachandran, G.N. Ramachandran. He passed away in 2001. Now, what I want to say, the possibility of this phi and psi angle depends on also for the side chain. If you have very bulky side chain, like I'm not going back to the slide again. Say for example, tryptophan, it has very big aromatic side chain. Then it will make more restraint here in the conformal angle of phi and psi angle. You can also imagine that if you have just glycine here as side chain, so forget about these other, like X1, gamma, or whatever mentioned, just forget about it. So if you have just hydrogen atom here, then it will not pose any kind of steric problem or any other problem. So as a result of that, this phi and psi will be more flexible. So based on that, this linear chain of polypeptide, whatever I draw here, it will try to fold in different way, right? It's kind of trial and error, right? And over the class, when we will discuss, you will get some logic and some discussion that will support that how these things are happening. And I will discuss some classical experiment also that actually primarily contributed to understand how protein forms. So here, Ramachandran plot, why I'm telling you, this plot at this level, you don't need to go in details, but you can see since it's an angle, phi angle and psi angle, as I told during the last slide. So the overall option is 360 degree, right? Minus 180 to plus 180, and y-axis also minus 180 to plus 180, where we plotted phi and psi. And here you can see again what is phi, what is psi. But what I would like to say, Ramachandran was studying in ISC Bangalore long time back, and he was in the Department of Electrical Engineering. Okay. So if I'm sure that electrical engineering students are also here, but you don't need to think about it. But after 1 or 2 years later, he actually thought that electrical engineering is not suiting well with him. So he came to physics department in ISC Bangalore. And at that time, in physics department, C.V. Raman was there. Okay, all of you know about C.V. Raman, right? So C.V. Raman actually wrote, head of this electrical engineering department, that your student is too bright for electrical engineering and he wants to study physics. This Ramachandran. So he is considered one of the most important scientists after post-independence. So then he actually admitted in physics department, and there he studied physics, and he was a physicist, and in the most of the his studies involving actually understanding protein structure, particularly biophysics. And then what happened, he contributed this thing that how what will be this angle and its kind of prediction. He was an actually experimentalist. He was synthesizing small peptide and carrying out experiment. And based on that, he decided that what kind of side chain or what kind of structure will permit how much of this angle, like phi and psi angle. Based on that, he made some kind of plot that is called Ramachandran plot. So, and from there he mentioned that if proteins are present in beta sheet conformation, that should be here. Their phi and psi should be somewhere here. Now you can ask me, sir, what is beta sheet? What is alpha helix? Yeah. So here that's the problem, that which one I will teach before. That's the problem. So please, for a little bit of time, you can ignore this, what is beta, what is alpha. But after 1 hour, you will understand what is beta sheet and what is alpha helix. But bottom line is, he contributed that that in a protein structure, what will be the permissible limit of this phi and psi angle. And most importantly, I would like to clearly mention, and we should be proud of that, that whatever he contributed now throughout the world, if any person, they wants to submit any protein structure, right? Protein structure can be determined by different technique. I told you most probably in some other classes, like for example, X-ray crystallography or NMR spectroscopy, or nowadays even more recent technique is cryo-electron microscopy. All are very advanced technique. But whatever you submit your structure, that should be validated by Ramachandran plot. That whether this phi psi angle and all these constraints are matching with his I would say theory here or not. If not, then you can even not, you cannot submit your structure. It's so I would say so right in that way. You cannot even submit your structure. So I'm just I will show you just one example here so that you will at least get that feeling here. You can see this, right? So there is one site, this is called PDB, that protein data bank. So all over the world, whoever solves some new structure, right? So they submit their structure in protein data bank. so that everybody can access the structure and they can work on it, and that will, that is freely available, site, and you will get the structure from there. So here, for example, structure of one molecule, say crystal structure of some protein, right? So here I am myself like also an author. So but any structure you can open, right? That is not a problem. But the thing is here you can see that Ramachandran out layer. You can see here, right? Ramachandran out layer with my cursor. So here this red side and this blue side. So here, so there are a lot of background data. I'm not going into that. But the thing is, it should match. So this structure intentionally I opened because this is statistically very sound structure. You can see this bar is very much close to this blue side. That means everything matches with this Ramachandran plot. It's not in the outlier of Ramachandran plot. If it is in the red side, that it's already in the outlier portion, and you have some problem with your structure, right? So this is his, contribution in understanding protein structure. And most importantly, also, I would like to mention that even if you open Wikipedia, you will find that Linus Pauling, Francis Crick actually mentioned that G.N. Ramachandran actually is capable of getting Nobel Prize. He didn't get it, but the category is same. But in 2001, he passed away. Unfortunately, he actually discovered how protein folds, and it's like, particularly this phi and psi angle. But last few years, he suffered from protein folding problem related disease, Parkinson disease, and he passed away in 2001. Okay.
Hello, sir.
Yes.
Sir, how can we define the angle phi and psi?
How you can determine, right?
Define, sir, define.
Define. So phi and psi. So in the, I'm just, I'm telling you, in the, like, even in the lecture note, I kept that because every time student they also ask. So here you can see, this is one one peptide plane here. This is the next plane. So this is basically torsion angle, right? Because this is the central carbon, and from here it's another plane. It's another plane. So basically phi and psi, those are torsion angle, or you can also say, because I'm telling you that plane, and also if you see like this two plane can be like this, can be like this. So you can also say dihedral angle, right? But in terms of definition, like, I would suggest you just go through it. You will understand because you have very sound chemistry knowledge also, right? So these are torsion angle, and also in addition to that torsion angle, I would like to say this since those two planes are joining there at the central carbon atom, this is also, you say, dihedral angle. And but what I tried to explain you, that this angle depends on the bulkiness of the side chain also, because we have 20 different side chain, and also you have to consider what is the next residue also, and how the side chain of that residue is bigger, or some charge present there, or some hydrophobicity present there. Based on that, overall folding depends. Okay.
Sir, I
Okay,
have
sir.
two questions related to the Ramachandran plot. Sir, for example, in the range of values of phi from 0 to 180 and psi from -180 to 0, there is no, like in the last quadrant, there is no protein structure given. So
Yeah.
does that mean that for those set of values of phi and psi, no protein will exist? Like that, those
No. Yes,
torsion.
you are right. Right, because as I told you, that little bit problem is whatever I'm telling you, that protein primary structure, but finally you will see proteins are not staying in a primary form. They make some secondary structure, that is kind of, one is called alpha helix, kind of spring-like structure, and one is like beta sheet, like strands. They are arranged in a stacking way, right? So here you can see in this slide, whatever you asked, you are right actually. In this, here you will not any value. Like whenever protein is in its alpha helix form, their value of this phi and psi should be somewhere here. And when they are in beta sheet, they should be here. And this is L is left-handed helix. Forget about this right now. This is not the natural one. Sometimes people can actually synthesize or make a left-handed helix, but that is not the natural one. As I told at the beginning, this is little bit of more details, but whoever more interested, I would suggest you that just go through it, and you will, particularly if you have more interest in, some of the students are from chemistry or physics department, if you pursue higher research also, even from engineering department, that is not a problem, but then I think you have to concentrate more on this thing. So Tanmay, I think the first question I answered, right? You will not get
Yes,
because
sir.
of this constant, because that part we didn't discuss yet. Because finally proteins are not present in a random way and they fold like this. Before that, they make some kind of secondary structure. So from primary to secondary, secondary to tertiary, and then finally quaternary. I will go through these steps. Okay.
Oh, okay, sir. And sir, in the beta sheet structure, that like it extends beyond the Ramachandran plot a bit, like in the upper portion. So, is that valid? Like,
1 minute. I would like to see.
yes, sir. In this red.
Yeah, I understand. So I have to see this image also from where I have taken. Your question is sometimes it go a little bit of outlier also, but that depends on, so most of the structures are actually like collected from X-ray crystallographic structure. So X-ray crystallographic structure in solid state, right? So sometimes it has a little bit of artifact also, right? All those structures are not from NMR. So NMR structures are from solution, like proteins are actually soluble in some solution. But X-ray crystallographic structure means you have crystallized your protein, and sometime you will get some like which is not completely native structure because of crystal packing. It's little bit distorted. So that is also possible, and we'll go through it. We will see actually. In many figure you will also see where you will see that it is inside the limit. But it's a good point. I will look into it. Okay, Tanmay.
Yes, sir. Got it.
Hello, sir.
Yes, Rajas.
So one small doubt that means from 0 to -180 exactly how are the angles oriented? Means how are the bonds oriented? I'm
Yeah,
not able
so
to visualize.
yeah. So it's a good question, but see, just to save the time, I'm not going into it. I'm just going to directly that slide. I kept that additional slide. So whenever you will see that later, with whatever I discuss, in addition to that, you will understand those things also, right? So that torsion angle, dihedron angle, and how you will see, like, so those things I actually I will provide at the end, like as additional study material. Okay, so then it will be easier. So I will like I'm just showing you little bit then because otherwise I cannot go there in the protein itself. This, but still after reading it will be easy for you. So for example, here on atom, here on atom, and here you have your eye. Okay, so you are looking this direction. Okay, so now how, like if you rotate this 30-degree, where you will see the position that way. So I have some animated slide. So I will actually attach those slides with this presentation before uploading into your MS Team. Okay, so then if you have more problem, then let me know through email. I will address your question. Because
Yes,
for
sir.
yeah, for if I need to go for all of the whoever present in the class, so I think I have to maintain some kind of like tidy, like whatever happening in other class, I should maintain that also. So anyway, here like here is some classes are like like some things are possible, some are not possible that way. Just some some just schematics here. So here you will see this is alpha helix and this is beta strand, but I will discuss how they form little later. Protein molecules are organized in a structural hierarchy. As I told you that when they are just attached by peptide bond, they are called primary structure. And from primary structure, they form the next order, that is called secondary structure. This is, so say first, for example, first thing, when you are getting primary structure, what is the driving force or what kind of bond? This is covalent bond, right? All of you know peptide bond formation, right? This is covalent bond formation. And the next is when you are getting the secondary structure, like whether it is forming alpha helix or beta sheet, this is nothing but because of hydrogen bonding of some main chain atom. I will discuss in more details that which atoms are involved in this hydrogen bond formation. So main chain hydrogen bonding, right? Atoms present in the main chain. Main chain means I am ignoring the side chain now. Main chain means just CONH, whatever is repeating there, whatever like that oxygen and hydrogen atoms, they are based on that this hydrogen bond will be formed. So I have detailed slide, let's just 1 or 2 slide later you will see that. Now after this secondary structure, still say for example you have this secondary structure here, right? And from this alpha helix you have this hydrophobic side chain here, right? Still they are unstable, right? Because I told you that hydrophobic structure should not be outside, right? So as a result of that, what will happen? So they will hold like this. That is called tertiary structure. So as a result of that, those hydrophobic side chain will go inside or packed inside in the core, and more kind of hydrophilic residue they will facing outside, like maybe with some charge residue, some polar residue, right? And the quaternary structure, this is not related to every protein. Only few protein they have quaternary structure. Quaternary structure means, remember, don't forget. So up to primary to tertiary, this is just single polypeptide chain. So you can see just one polypeptide chain. You have started with N, that means N terminus. I told you what is N terminus. Here C terminus, right? And now you get this alpha helix and beta strand or beta sheet. Then you get the tertiary structure. That means it's the same, just one polypeptide chain, right? But in some protein, like multiple polypeptide chain, they stay together, and particularly they function just, they carry out a single function. That means those multiple polypeptides should stay together in order to achieve their function. Example here, this is hemoglobin. Hemoglobin. So hemoglobin have 4 polypeptide. 4, see 1, 2, 3, 4 in different color, right? So 4 polypeptide. So can you give another example? Like hemoglobin, already I told that example of a quaternary structure of a protein. I actually discussed that before. Any example? I would like to see whether you understand the like overall idea of this primary, secondary, tertiary, or quaternary.
Sir, RNA polymerase.
Very good. RNA polymerase. Because I told you, right, that alpha, beta, beta prime, and that sigma, all those things work together, right? So as a result of that, you are right. So RNA polymerase, that is one example I already discussed. That is an example of quaternary structure of a protein, not just single polypeptide there. Multiple polypeptide, they stay together and they work together. Now, secondary protein structure. So mostly whatever I will discuss today, this is someone who is actually in the chemistry class, either in IIT or any chemistry honors in any university. This is very standard, like one portion, like at least 50 marks or 100 marks should be biochemistry. And in biochemistry course, protein chemistry is the very most fundamental one. Without protein chemistry, you will not pass biochemistry, or you will not understand biochemistry also, right? So because in some courses I have seen, they sometimes ignore nucleic acid or other things, but protein chemistry is everywhere. So now if you see, like as I told you, that this is just alpha helix and beta sheet, but here characterized by main chain NH and CO groups participating in H bond. But here still you will not understand the details. In the next slide I mentioned here. Here very carefully listen or see what is happening here. So as I told you that in a polypeptide chain, in a primary structure, you have N terminus, right? Amine group here, and here carboxy, or this is called C terminus, right? So just we are showing a small segment of a polypeptide chain, and this is one amino acid here, like its number as N, and this is N plus 1. It's already written in the down also. N plus 2, N plus 3. That way, the repeating, or I would say like multiple amino acids are attached by peptide bond, and you can continue in this direction more and more, right? So as a result of that, you can see that here we can ignore this side chain, because I already told that when we are discussing about the formation of like alpha helix and beta sheet, that is secondary structure, that is based on the main chain hydrogen bond. But those side chains are also like, they are involved in a different way, that how big the side chain is, whether that will creating some problem or not in order to get that kind of helix. So that I will come little later. So now if you see in this amino acid, right, here you have the CO group, right, carbonyl carbon here, and this is oxygen here, CO group. And now here in the next one you have NH group, and it's repeating like this. What happened in case of alpha helix? That means you have one polypeptide like this, but in order to form the alpha helix, certain kind of like specific order of hydrogen bonds should be there. Okay, that I am trying to explain you. The specific order is that hydrogen bond between N, at the N position, this oxygen and the N plus 4. So this is N, then 1, N plus 1, N plus 2, N plus 3, N plus 4. So as a result of that, it will form hydrogen bond with this hydrogen, this O and H here. See, again, next amino acid here, again you have the oxygen group, right, CO. This oxygen, this will form again hydrogen bond with this NH group. Again this one, right, it will form hydrogen bond with the next one, which I didn't show here. So that way, a specific order of hydrogen bonding will continue, right? So as a result of that, this N plus 4 hydrogen bonding between this CO and NH group make this alpha helix. Now I would like to ask you one simple question, that tell me what is the distance between, or distance of hydrogen bonding, approximate. I'm not like, it's also depend, not always constant. So what's the distance of hydrogen bond? You heard that, like you
3
studied.
angstrom.
Huh?
3 angstrom.
Yeah, 3 angstrom. You can say 3 angstrom, right? Very good. So 3 angstrom. So like, in protein structure, if you see 2.8 angstrom, 3 angstrom, 3.1 angstrom, but approximately like, in ballpark, it will be 3 angstrom. So now, if you see what I told, like to all of you, I want to say, I told that the CO of this N residue that makes hydrogen bonding with N plus 4. This is far away, 4 amino acid away. How that can be 3 angstrom? It is not possible, right? So the only thing is how then this hydrogen bond, specific order of hydrogen bond will be formed, right? So then you have to think about some kind of spiral, right? So that although this is N plus 4, but that fourth residue is not far away. This is much closer. How you can imagine that? If you can make an imaginary axis, or you can make some kind of coil, right? So as a result of that, you will see that residue, that N plus 4 residue is actually, they are close to each other and they are in the range of forming hydrogen bond, right? So that way, this alpha helix form. Do you know who actually provided this concept or discovered this alpha helix? Any idea? Not in protein chemistry or biology. Maybe you studied in chemistry. Linus Pauling, he got Nobel Prize, right? Linus Pauling actually solved this puzzle of alpha helix, how alpha helix form, and these are the this. Here I want to mention here, whatever I actually discussed before, like molecular biology part, the replication, transcription, translation, those are more recent. But understanding amino acid, peptide bond, peptide, those are more they are discovered and people used to work even before that. before understanding of this molecular biology, like DNA to protein, like RNA to protein. Protein was known before, but thing is that after the discovery of this transcription, translation, everything, they connected it together. That proteins are the result, or they are coming from DNA, right? That was not known before. Right, that was not known. Protein was known, like, people actually used to work with protein like in 1930, right? But in 1960s, like 50s, 60s, that time we know about like more about like DNA, RNA, their structure, this molecular biology, whatever I discussed in previous classes, right? But proteins, like understanding proteins, those things are much older. But the link was not there, that how proteins are getting generated, from where this information coming, that link was not there before. But whatever we are discussing, we started in that way, that from that link we are going, right, from DNA to RNA, RNA to protein. That way we are moving forward.
Excuse me, sir.
Yes, Rajas.
Sir, that in the alpha helix, it's not proper arcs. No, it's because due to the bonds are straight, so it's not proper arcs.
Proper?
Arcs of circle means
Okay,
not
okay.
proper helix.
Yeah. So let me one minute. So I didn't show you the detailed structure yet. Just a minute, Rajesh. Here. So here, now see in a more details way. So in, like, it's kind of, I forgot to bring this. So this is the imaginary axis, and this polypeptide, linear polypeptide chain is moving like this. Okay. So this one is right-handed helix. So similarly, as I told you, right-handed means just, take your right hand and thumbs up, and then whatever this, whatever this arrow is going like, so this is your right-handed helix. And now here, in case of DNA, what happening? The two strands are going like this, but in this case, not two strands, just one polypeptide chain, right? And now, if you can see here, that in this case, this hydrogen bond, they are nearly parallel to the axis, and also some informations are there, that this is because of, we know the structure of alpha helix. There are many protein structure present in PDB. You can open that in your computer and you can see how they look like. So based on that, every 3.6 residue makes one turn. That means in one turn, in 360-degree, you have 3.6 residue. So as a result of that, if you consider the central carbon, like, so the next central carbon should be, what will be the angular distance between two alpha carbon atom? What will be the angular distance between two central carbon atom?
Sir, 100 degree.
Yeah, 100 degree, because here you can see, as I mentioned this one already, that 3.6 residue make one turn, right? So as a result of that, 100 degree, like, and the next alpha carbon atom, just the angular distance I'm saying, and that way alpha helix is going on, right? So now in a protein,
Excuse me.
yeah.
Sir, 3.6 residues means what? 3.6 polypeptide chain.
3.6, not polypeptide chain. Polypeptide chain is just one polypeptide chain. 3.6 here means amino acid.
3.6 amino acids.
Yeah, 3.6
Okay.
means like as in DNA also you like studied like 10.4 per turn. So this is just some information that like 3 residue cannot complete the full turn, like 360 degree, and 4 residue little bit extra. So it's kind of, by calculation, this is 3.6. Got it, right? If you see here in the figure, so here, see, this is the central carbon. Yeah. So this is the central carbon.
Sir.
So this purple color thing, those are the side chain. This going like this. This is the main chain, right? Going like this, and then again it's making like this kind of helix. And here, like 3.6 residue per turn. That means whenever 3.6 means nothing, you cannot like divide this residue in that way. But when this same amino acid is continuing, by that 360 degree is covered. That's why we are saying 3.6 residue.
Sir, how can you measure amino acids in fractions, sir?
How you can measure? You can measure. Actually, I'm here. So
sir, we can
yeah,
like three carbon chains. Like
huh?
so the first oxygen chain can be like there, and like after 3 counts, there were 5 of those members. We can divide it into.
Yeah. That is 3.6. I understand his question also that it depends. Like so, like how why I'm telling 3.6. That if you see the main molecule, like main chain, they have C O N H. That way it's moving, right? So the thing is, this is I will suggest you Sarir Hanan. So just if I get time, whatever I actually started with this structure, like so if I open this in PDB, then you will get like I will be able to answer this question. So we can open this structure actually, right? I'm just showing you just a minute. Then it is like many of you are interested, but the thing is, 1 minute. Okay, so let me, when I will take break, that time I will download one of those structure. So that way it will be easier. Okay, because not downloaded in my desktop. So then it's more like whatever I am just discussing, those things are also like written here in the legend section. So now if you go in the next thing, the beta set. So here number of beta strands are involved. Beta strands means here in this case this is little bit different. In this case you will see this, if this is your N terminus and C terminus, what happened that you started with here N terminus, then it's again come back like this, again come back like this, this is C terminus. Just I'm giving example. Now hydrogen bond formed between residue present here, right here and here in this strand, like in the here. So as a result of that, this segment, this is called one beta strand, this segment another beta strand, another beta strand. So multiple beta strands, nothing but beta strand means just like primary structure, but they are arranged in such a way that finally they present like this and they form hydrogen bond, and because of this specific hydrogen bonding they are staying like this, and together this is called beta set that is constituted by multiple beta strands. So here you can see like this is going like this, and then again you have hydrogen bonding, and then again another strand, again hydrogen bonding, but those should not be discontinuous. In this figure it's discontinuous, but it should be, say for example, it can be like this. Then again you might have one alpha helix. Okay, again it's coming back, again another beta strand. Also possible, right? That's why protein structures are very complex. Another alpha helix, again it's coming back, again beta strand. They are forming beta strand. Okay, so there are different types of fold and exist in nature, but that means proteins, they can made up of like completely alpha helix like hemoglobin. Some of them can be completely beta set like some of the for example antibody. All antibody, they are made up of only beta set, and some of them they can have both alpha helix, beta set, everything
Excuse me,
together.
sir.
Huh?
Sir, at the turning point, there is hydrogen bond or covalent bond
Turning point,
on
very good
beta.
question. The turning point, most turning point, covalent is already there everywhere, like that is peptide bond, right? Covalent is already there. But those turn, that is called beta turn. Intentionally, I didn't discuss in that beta turn. That is not neither fitting with the alpha helix or neither with the beta set. That is in protein chemistry we say that is either loop or random coil. That is just some flexible, like normally 4 to 5 residue or sometimes 7 to 8 residue, but that's a random coil. Neither they are giving you like, proper alpha helix structure or beta strand, nothing. Right? Those are the connecting region between two alpha helix or two
Sir,
beta
I
strand.
am asking about bonds, sir.
Bonds, nothing.
Hydrogen bond or covalent?
No, hydrogen bond, like hydrogen bond, if the hydrogen bonds are present in the random coil, what do you think? What kind be the residue? Mostly polar residue should be there. So as a result of that, they might, interact with the water molecules through hydrogen bond. But major bond is nothing but peptide bond there, right? And in a folded structure, some of the side chain, like for example arginine, they have positively charged side chain. They might have electrostatic interaction with another residue present in the protein itself. Okay, but they are not following any specific order of hydrogen bonding, what I told in case of alpha helix and beta sheet. Otherwise, they will also be part of this alpha helix and beta sheet, right? So that's why they are called random coil, right? So let me go through it. Then when I will discuss the overall like tertiary structure, then you will get this point in a better way. Now, so here like two things mentioned here, parallel and anti-parallel beta sheet, and then I will take some break. Okay. So parallel beta sheet, anti-parallel beta sheet. So tell me, I know that none of you read about this. Without searching Google, like based on my whatever we discussed together now, can you some of you can you try to answer what could be parallel and anti-parallel beta sheet? Please don't do Google. Without that, just try to think and try to answer.
Sir, if N and C are present on same side and
Yeah.
different side on that basis.
Yeah. But how it can happen? You are right.
You told me one alpha helix example in that C
Yeah,
was on
alpha
other
helix
side.
or yeah. You are very close, or you are absolutely right what you are telling. I'm just giving you just an example, right? So also you should remember that protein synthesis, always from N terminus to C terminus, right? During translation. So like this. Now this, if this protein can form beta set, I'm just giving you some to explain what is antiparallel and what is parallel. Just if you consider this is a linear chain of polypeptide, our primary structure. Now it can hold something like this, and then some loop, and then again like this, and then again loop, again this, again loop, and here you have C terminus. Okay, so this portion is loop. They are not following this specific order of peptide bond for hydrogen bond formation, but here you have specific hydrogen bonding. Okay, so now if you see in this segment, what is N and C? This is N terminus and this is the C terminus. In this segment, again it is going like this. Here, what is in this segment, what is N and C? If you just cut it imaginary, then this is N and this is C. Again here in this segment, N, C. Here, N, C. So as a result of that, this is anti-parallel beta set. But another way it can fold, that is also present in nature. So this is N terminus, right? This is one strand. Then this loop somehow went away. Okay, they are forming maybe one alpha helix here, as you told, one student told, and then forming some loop again coming back here like this. And again, this is another segment, right? This is another segment. Again, it is going like loop and forming another alpha helix. Like this is again the C terminus of the protein. So now if you consider this small segment here, right? This is N terminus and this is C terminus. And then it is going like this, going like this. Again, this is N terminus and this is C terminus. Again, it is going like this, N terminus, C terminus. So this is parallel beta set. Okay, I hope that you understand.
Due to the due to the order of N and C, like in the antiparallel it was NCC and NCCN, so that's why it was antiparallel, and here It is always NC
Yeah,
and CNC.
yeah, exactly. So those are, I'm just writing there, right? Because if you see the now, if you see the structure, right? I'm just showing you here. See, anti-parallel here, and here this is the direction N to C, and here parallel, all are in same direction. So both are like abundantly available in nature in normal condition, and because of their orientation, if you see in anti-parallel in anti-parallel structure, again you will see that how you will determine this. It like you need little bit of more protein chemistry, and you have to see some protein structure. Because of their orientation, if you see this hydrogen bonding is completely, like I would say, perpendicular to the axis. Here, see here, little bit of angles there, right? So I'm not going into that much of details. And another thing I would like to mention here before taking break. So here, in case of antiparallel beta strand or beta sheet, mostly those strands are almost continuous portion. Like here only you have in this portion, this turn, only you have 3 to 4 amino acid, that is enough. It might have more, but 3 to 4 amino acid is fine actually here. But in this case, you have like lot of extra residue and coming back here. Then it's from the next beta strand. So as a result of that, antiparallel beta strand are kind of in a more continuous region of a primary structure. But in case of parallel beta sheet or parallel beta strand are some kind of discontinuous portion, they can come back from come from a different portion of a polypeptide chain, and finally they fold together. But now the puzzle is, as I told you, that protein folding people used to study like 40 years back, and even people are studying now, and this is one of the most common biological field. But biology that is being studied in various departments across the various department in physics, chemistry, biology, engineering, even in your IIT, I would say, even you in IIT Kharagpur, if you see, even in computer science also department, you will see some people they are working with like protein structure, simulation, and all those stuff. So this protein folding, because the idea is the overall understanding, although it's clear that how it is getting folded, but who decide that, how it has been evolved, those answers are not known, right? And people are trying to answer those questions. And in order to answer those questions, honestly, you don't need to know a lot of biological details. That's why it is more kind of interdisciplinary, and people are working on it also. So that is another reason. But let me see what is the next slide. I will take break. So yeah. Yeah, so I will take actually break here, just 3 to 4 minutes, and then I will come back and then discuss. Okay, so please take a break and then come back within after 3 minutes.